Welcome to our webinar, How Companies Are Developing Next-Generation Biomaterials for Orthopedics. Our moderator is Matt Pogge, Senior Vice President of Regulatory and Quality at Acuitive Technologies. He is joined by Dr. Richard Tran, Director of R&D at Acuitive Technologies, Sriram Sankar, Director of R&D at Diffusion Technologies, and Dr. Mary Beth Schmidt, Vice President of Research and Development at Hyalex Orthopedics. Our sponsor is Precision Medical Technologies. Precision Medical Technologies is a contract manufacturer of orthopedic implants and instruments with a focus on spine, extremities, trauma, and sports medicine. The company's 3 separate instrument, implant, and disposable divisions allow it to focus technical resources that drives high-quality, cost-effective production.
Over the next 30 minutes, you will hear from 3 scientists who will share information regarding new materials technologies that are under development and are moving into commercialization. Each presenter is a key contributor in a young, semi-startup organization. All are looking to address an unmet clinical need, to succeed where others have not, and to seize a market opportunity. A common goal for all is the intent to provide a biomaterial that contributes to the healing process, not merely to provide an inert biocompatible scaffold for host tissue to form upon. The technologies differ, but each face similar challenges in the discovery and development phase and on the path to regulatory clearance. Following the presentations, there will be a brief question and answer period. I'd now like to introduce Dr. Richard Tran. He will discuss a new bioresorbable material named Citrogen that has been developed by Acuitive Technologies.
Thank you, Matt, for that introduction. I'll be talking today about Citrogen orthopedic biomaterials. So if I had to define Citrogen, I would call it a next-generation platform biomaterial. Its modular design allows it to be modified for a wide variety of applications, and it's all based off the citrate molecule. The citrate molecule brings many material benefits and also biological benefits. Citrogen is a thermoset bioresorbable elastomer, meaning it's much different than the traditional thermoplastics that are out on the market today. It's elastic in nature, and it resorbs without chronic inflammation or acid dumping. And it's purposely designed to be functional and bioactive, all to instruct tissue regeneration. The design inspiration behind Citrogen was inspired by nature, namely the extracellular matrix. The extracellular matrix is a protein network of collagen, elastin, proteoglycans, all cross-linked together to stretch and recoil in dynamic mechanical conditions. It all regulates cell survival, adhesion, proliferation, migration, and differentiation. So in looking at the extracellular matrix structure-property relationship, we wanted to select a molecule that was multifunctional. Multifunctional so that we could use the molecule to increase chain length and also preserve pendant chemistry. The preservation of the pendant chemistry allows these polymer chains to be cross-linked together, similar to the way the extracellular matrix is designed. Citrate also, in addition to biomaterial design, offers many biologic benefits. It's traditionally known to be a key contributor to the Krebs cycle for energy production. But when you look at the tissue citrate levels, you can see that there's almost 400 times more citrate in bone and teeth than soft tissues and blood plasma. Researchers have known that citrate levels were high in bone for a while, but it wasn't until 2010 and 2014 in 2 separate articles published in PNAS where they elucidated the role of citrate in bone physiology. One group in 2010 noticed that the citrate molecule was found bound strongly to the surface of nanoapatite, and it restricted the apatite growth in one direction and allowed it to grow in another direction, forming plate-like structures. A second publication in 2014 verified that the nanoapatites were actually bound together through a hydrated layer of citrate, giving bone its unique properties. So looking at the material design now, once it's composited with ceramic, we're looking at these compressive stress strain curves here. You can notice that traditional thermoplastics begin to yield and permanently deform under 10% strain, whereas Citrogen is an elastomer, so there is no plastic deformation and it just fails up to about 50% strain. Now this is loaded with up to 60% ceramic. So it has a high ceramic content and still maintains elasticity. Looking at the pictures on the bottom, you can see the Citrogen material on top, where it can be compressed up to about 20, almost 30% strain and recover without permanent deformation. In comparison, you can see the PLDLA on the bottom, and after 10% strain, it starts to permanently deform. Compressively, Citrogen is very strong. It's stronger than cortical bone. But what's interesting is that the modulus of the material is very soft. Its modulus is more comparable to cancellous bone. Citrogen also has some bioceramic interaction due to the citrate molecule. You can see SEM-EX pictures here where calcium's stained in blue. And you can see the Citrogen material has a very uniform bioceramic content with up to 60% to match the native bone content, whereas PLDLA TCP materials have agglomerations of TCP and bioceramic and can only be loaded up to 30% before becoming brittle. Citrogen's life cycle by day 1 starts to elute essential molecules such as citrate, calcium, and phosphate to the surrounding tissue. By mid-stage, polymer resorption is still continuing, molecule elution still occurs, but it leaves behind an HA construct. Normal tissue healing occurs and the implant is then ultimately metabolized. Late stage, their normal resorption occurs and normal physiology is expected. I mentioned on the last slide that citrate is released. A recent study in PNAS in 2018 showed that exogenous citrate is uptaken by human mesenchymal stem cells through a specific citrate transporter. Once citrate is released and enters the human mesenchymal stem cell, the internal citrate levels go up, as does the ATP or energy. Looking at the graphs on the right, you can see the intracellular citrate uptake, where it's normal culture media and the relative citrate amount, and then once citrate is added to the media, how much the internal citrate levels increase. Next, they looked at stem cell ATP production, and control cells showed normal ATP production, but once citrate was added to the media, it was a significant increase in the amount of ATP production. Once the citrate transporter channel was blocked, the ATP production then returned back to normal. Looking at stem cell response, this is human mesenchymal stem cell osteopontin production. This is a late stage protein required for bone remodeling at 7 days. Here are stem cells grown on PLDLA TCP. What you're looking for is a green stain for the osteopontin. It is here, but it's barely visible, whereas the citrate material, the Citrogen material, shows very nice osteopontin and a lot of osteopontin production. What was very interesting in this study is that usually you would see this in osteogenic media, meaning media that's been tailored to differentiate the stem cells, but we also saw this when we were just using normal media. So that means that the Citrogen material, the molecules that are being released, could influence stem cell differentiation. Next, we wanted to look at the biological evidence using a sheep study. We looked at porous plugs implanted in the vertebral body. You can see the Citrogen material here. It's very faint because the high ceramic content matches the bone density. We looked at it 1 month and 6 month histology. We saw evidence of biological ingrowth and biocompatibility. By 6 months, tissue had infiltrated the interior of the pores, and it was absent of any signs of chronic inflammation or any fibrous tissue formation. The orthopedic applications that we are targeting for Citrogen, we've gotten 3 510Ks. The first one was on a Citralock, which is an interference screw for foot and ankle and ACL repair. The second one was a Citrafix or a suture anchor for the foot and ankle and also shoulder applications. We are working on some new products in the pipeline. We're working on, the first one is a shoulder scaffold. So it's a 3D printed porous construct used as an interpositional scaffold to help regenerate the natural enthesis. Another is a Citra wedge for foot and ankle. And also we're working on a very porous foam-like sponge we're calling Citra foam as a bone void filler. Some of the longer term projects we're working on is a in situ cross-linkable version of this, where it would be a biodegradable bone cement-like material. I mentioned earlier the modular design about Citrogen and why it's a platform material. This is a busy slide, but you can see that it all starts with the citric acid molecule. We then select hydrophobic or hydrophilic diols to control the degradation rate. And because of the unique bulk chemistry of the citric acid molecule, we're able to attach many different properties or molecules to exhibit different properties. We've talked a lot about composites, but you can add molecules with catechol groups and make bioadhesives out of this material. We add vinyl groups to create in situ cross-linking abilities, ascorbic acid for antioxidant effects. By reacting any of the amino acids to citrogen, you get a very unique fluorescent mechanism or property. And with hydrophobic domains, larger dials, you get shape memory effects. And then introducing urethane chemistry or click chemistry, we can significantly increase the mechanical properties of citrogen. And the very unique part about citrogen is that it's not just one aspect. You can have any combination of these to fit a particular application. So you could make composites that are adhesive, or composites that are in situ cross-linkable, or you could have a fluorescent material that is also an antioxidant. So it's a lot of things that can happen because of the modularity. And as people do more and more research, we're discovering more and more molecules that can actually introduce unique design aspects. So in summary, citrogen is a citric-based platform biomaterial. It's a bioresorbable elastomer, not a thermoplastic. It's biomimetic in that it mimics the extracellular matrix structure. It has unique bioceramic interactions and allows us to incorporate up to 60% ceramic to match native bone content. It has dynamic mechanical benefits, and it's a metabolic molecule. It resorbs without chronic inflammation. It resorbs without bulk degradation or any acid dumping. pH stays neutral throughout the life cycle. And the modular design can accommodate a wide variety of applications. And at this time, I'd like to acknowledge some of the labs that we're working with: Penn State University, Northwestern, University of Ireland, University of Pennsylvania, and Colorado State University, and then all the team members at Acuitive for helping me put this together. And thank you, OnTech.
Thank you, Richard. We'll now hear from Mary Beth Schmidt from Hylex Orthopedics.
Thanks, Matt. As Matt mentioned, I'm the VP of R&D at Hylex Orthopedics, and I'm going to be talking to you today about a unique biomaterial called Hylex cartilage that we are developing to treat painful cartilage lesions. Our company mission is to develop a transformational synthetic cartilage technology and implant systems associated with this technology to treat diseased and damaged joints. Our goal is to address unmet clinical needs and improve patient quality of life. Hylex Orthopedics was founded in 2016, and we're located right outside of Boston in Lexington, Massachusetts. At this point, we've raised more than $33 million in Series A funding, and we have a strong and supportive group of investors and partners who've been instrumental to the progress of our company. Our clinical advisors include some of the top orthopedic and sports medicine surgeons in the country. We're currently at the preclinical stage in the development of our first product, led by an experienced team with deep expertise in the medical device industry and orthopedics. I want to begin by providing you with some brief background on the clinical problem that we're addressing with our technology. I'm going to use the knee as an illustrative example in my discussion, but this discussion could apply to other joints as well. Hyaline cartilage is the specialized tissue that covers the ends of our joints. It's that smooth, tough tissue that provides a very low friction interface and allows the joints to glide freely, even under very high loading. It's able to do this because it's a biphasic material with a high water content. Some of the water is trapped in the tissue and it gives it its compressive stiffness. The rest of the water is mobile, so it can move in and out of the cartilage under compressive loading. It also plays a role in lubrication. When loads are removed from cartilage, water moves back in. So cartilage can become damaged through traumatic injuries, which can create painful and debilitating lesions that can extend even into the bone. Since cartilage lacks a blood supply, it's unable to heal effectively on its own. As a result, untreated cartilage lesions can degenerate over time and lead to pain and loss of function. Currently in the US, patients with cartilage lesions have relatively few options for surgical cartilage repair. The surgical standard of care consists of debridement with or without a microfracture procedure. Following these surgical interventions, patients might feel some improvement, but pain relief is often temporary. There are specialty procedures that may be performed for larger cartilage lesions, such as a cartilage cell or chondrocyte implantation, which is a procedure that requires 2 separate surgeries. Another alternative is transplantation of cartilage from other areas of the patient's own joint or from a cadaver joint. Both of these approaches require specialized skill for the surgeon to perform, and they require long patient rehab, frequently months before even returning to normal activity, and up to a year before returning to sporting activities. The outcomes from cartilage regenerative techniques like these are variable, and they depend on factors such as patient's age, weight, and their activity level. So we all know someone with knee pain, myself included. In fact, knee pain affects 1 in 4 adults in the United States. Over 1 million patients in the United States are diagnosed each year with knee cartilage lesions, and about half of them undergo some type of cartilage repair surgery. Cartilage lesions also occur in other joints, such as the hip, the shoulder, the ankle, and even the big toe. Patients are aware of the poor long-term results from procedures like microfracture, which I showed in the previous slide. They're beginning to decline this type of surgery, and they're looking for alternatives. This is the unmet need that Hylex Orthopedics is targeting. At Hylex Orthopedics, we've developed a patented material platform that's intended for use for repairing painful cartilage lesions in joints. Hylex cartilage is the first synthetic bearing material that's been designed from the ground up to mimic the critical properties of hyaline cartilage. So on the left side of this slide, you see an illustration of the key components of hyaline cartilage. You see collagen, which gives the cartilage its strength, and proteoglycans, which are these large bottle brush molecules that are negatively charged. The proteoglycans are what attract the water into the cartilage, and then the water is able to move in and out under cyclic joint loading. Similarly, Hylex cartilage, shown on the right side, is also composed of a strong polymer network, shown in green and labeled as polymer A in the right diagram. And this is combined with a second negatively charged polymer, polymer B here, shown in pink, that draws in the water. This water is also able to move in and out of the material in response to loading in a similar manner to native cartilage. On the far right-hand side, you see a sample of Hylex cartilage. Hylex cartilage has a gradient structure, so it's got a very smooth, lubricious, hydrophilic bearing surface, and then it's got a hydrophobic back surface to allow for attachment. The size and shapes of cartilage implants that we can make with Hylex cartilage can vary and depends really on the application. The Hylex material platform also includes an adhesive that we developed specifically for the fixation of Hylex. The adhesive forms a strong, permanent bond with the bearing, allowing a wide variety of implant designs to be possible. Unlike other polymers like hydrogels, Hylex cartilage is formulated to be stronger than the native cartilage so that it will withstand the millions of loading cycles experienced in a joint over time. As the figure on the right shows in this slide, the material strength in tension, shear, and compression all exceed the properties of healthy native cartilage. This is important in a joint like the knee, which has complex motions that involve both rotation and translation. Knee loads exceed body weight by several times just when walking. Other activities such as climbing and jogging result in further elevation of these knee loads. When we consider the compressive stiffness, we know that traditional orthopedic materials such as ultra-high molecular weight polyethylene and cobalt chrome that are used for hip and knee arthroplasty implants are hundreds and even thousands of times stiffer than healthy cartilage. And despite this difference in the stiffness, these materials have been tried as cartilage repair implants. And then as the inset photo shows, the mismatch in the properties can result in long-term irreparable damage to the opposing cartilage surface. In contrast, the compressive stiffness of Hylex cartilage is very similar to native cartilage. This allows loads to be transferred to the surrounding and opposing cartilage in a more physiologic manner, helping to preserve the joint tissues. Because its design mimics cartilage, Hylex cartilage has an extremely low coefficient of friction, similar to the cartilage in a normal healthy joint. Coefficient of friction measurements of various combinations of cartilage and Hylex cartilage are shown in the figure on this slide. Both Hylex cartilage articulating against itself in the orange bar and Hylex cartilage articulating against normal cartilage, the light blue bar, have very low friction coefficients, similar to cartilage in healthy joints. In contrast, cartilage tested against a polished cobalt chrome surface has a coefficient of friction that's about 10 times higher. This slide illustrates the value of our platform technology for the development of a wide variety of implant solutions to treat painful cartilage lesions. The ability of Hylex cartilage to be used for implants that articulate against native cartilage has been confirmed in a standard rabbit trochlear groove model. In this study, Hylex cartilage was implanted in the groove, contacting the opposing cartilage surface on the patella. A non-lubricious polymer was used as the control. The center left photo shows the surface of the patellar cartilage that articulated against the Hylex cartilage. India ink staining was used to highlight any cartilage damage. You can see that the surface looks pristine with no damage visible. And this is also confirmed by the histology section on the left. In contrast, the center right photo shows the patellar cartilage after articulating against a control polymer. The staining highlights the damage onto the cartilage surface, which is also seen in the histology section on the right. Unlike traditional orthopedic implant materials, Hylex cartilage, with its cartilage-like mechanical properties and its low coefficient of friction, can be used to repair cartilage lesions and preserve the opposing cartilage surface. So as you can see from the data that I've presented here today, Hylex Orthopedics is developing a transformative orthopedic materials platform with applications for joints throughout the body. Compared to the surgical cartilage repair and replacement options that are available to patients today, a synthetic cartilage implant may allow treatment for a broader range of patients, including those who might not be ideal candidates for biologic treatments. Our experienced team is focused on bringing this new technology to market and being the global leader in cartilage repair. And then to close, I want to close on a personal note. I've worked in orthopedics and specifically cartilage and wear and tribology for more than 25 years. And as I said, I am a person with knee pain. I've helped Numerous companies bring new innovations to market, but Hylex cartilage is by far the most exciting technology I've seen. I look forward to the opportunity to help bring this new approach to treating cartilage lesions to patients across a wide variety of application areas and helping those patients be active and stay active. Having completed the standard preclinical and bench tests, we're excited to be going into the clinic next year with Hylex cartilage. And in preparation for this, our company is growing with several openings on my team. So I want to use this forum to encourage people to reach out to us and find out how you also can be involved in this groundbreaking technology that is going to transform orthopedics. I want to thank you, Matt, and thank Omtech for the opportunity to speak today.
Thank you, Mary Beth. We'll now hear from Shriram Shankar from Diffusion Technologies. Thank you.
Thank you, Matt, for that introduction. And today I'm going to be talking about understanding the relationship between osteoimmunology and bone response of orthopedic biomaterials, and specifically touch upon Z-Fuse, a technology which harnesses this relationship between osteoimmunology and bone response. I'm Sriram Shankar, the director of Diffusion, and I want to acknowledge our collaborators at the Department of Neurosurgery at Allegheny, as well as the McGowan Institute of Regenerative Medicine out in Pittsburgh. So this is our humble abode here in Austin. We were founded in 2009, and we have been dedicated to engineering and developing a set of orthopedic biomaterials which have immunomodulatory, antimicrobial, and tissue healing effects. And we are also an end device manufacturer who are primarily focusing on the orthopedic and spine market in particular with respect to fusion systems. So in the spine biomaterial field, we have two basic materials. One has been titanium, and titanium, as we all know, traditionally is a bone-friendly material, but in the spine, when it comes to load sharing, again, the modulus of titanium hasn't been ideal, as well as the fact that from an imaging perspective, titanium has been slightly problematic, especially in terms of evaluation of fusion, has been an issue with titanium, as well as the inability to do revisions in case of revision surgery being needed. So that brought about the advent of PEEK in the market about 15 years ago. And PEEK really, had a stronghold in the spine fusion market. And what they found was PEEK's radiolucency and biomechanical load-bearing characteristics was pretty much similar to cortical bone, and that was something that was a big plus. Another aspect of PEEK was its radiolucency, and from a visualization standpoint was such a big positive. But what has happened in recent times is, well, clinically, more and more cases have come about wherein we see this fibrous encapsulation around PEEK. And this is something that is, kind of perturbed the surgeon community and has led to a pendulum shift back towards titanium with respect to a fusion material. And one of the things that we see around PEEK is this fibrous encapsulation. And a paper by Renee in Spine kind of elucidated the mechanism. She laid out the fact that MSCs don't differentiate into osteoblasts on PEEK surfaces, and factors produced by MSCs on PEEK surfaces favor fibrosis, which is what we see clinically. And also we know that pro-inflammatory cytokines like interleukins have a role to play with fibroblast proliferation and synovial pannus formation. So quickly getting into our solution, which is Z-Fuse, which is the primary focus of my talk today, is Z-Fuse is basically a PEEK and zeolite composite. Zeolite's nothing but aluminosilicates, and they basically, when impregnated to develop this into PEEK, to develop this composite, not a coating, result in very different properties that I shall touch upon. And that's something that we've developed for the US market, and we've recently gotten approval in 2019 for it. To the right, you see CleanFuse, which is, again, these zeolite molecules can be loaded with different cations, in this case, silver, and we have actually elucidated a mechanism wherein we can release low parts per billion of silver such that it takes care of any sort of planktonic bacteria, but at the same time helping with the bone response. And so that's something that's out in the European market from about 2015. So these are the two major materials that we've gotten approvals for, while we have a few more products in our pipeline. But Z-Fuse will be the focus of my talk today. And so again, like I said, Z-Fuse is made of zeolite and PEEK. Zeolite, like I said, is aluminosilicate. It's negatively charged, super hydrophilic. And you can see that zeolites have been shown in literature to increase appetite formation, enhance osteoblastic proliferation, ALP activity, as well as synthesis and secretion of TGF-beta. So one important aspect that we want to understand today is what is the correlation between osteoimmunology and bone formation. And we all know that biomaterials can elicit a negative immune response, and that's something that we test for through 1099-3 testing. But what we don't know or understand very well is, can a biomaterial elicit a positive immune response? And if so, does that affect downstream outcome? And is it potentially due to material chemistry? I think those are the kind of answers that the Z-Fuse technology was looking at answering and understanding, and we shall touch upon that. So when you look at macrophage response in the body, generally the monocyte enters the tissue or site of injury or insult and turns into a macrophage. And there are cues like interferon gamma and lipopolysaccharides, which result in the macrophage actually shuttling into its classic M1 phenotype. Now, a classic pro-inflammatory M1 phenotype is what you generally see immediately, after insult or injury. And this is actually an important state, because what it does is it actually brings the other actors of wound repair to the site of injury. So this is something that is essential and needed at the start. But what we have come to understand is in recent times that these M1 macrophages or pro-inflammatory macrophages actually quickly shuttle into a pro-reparative state called M2 state. Now, phenotypically, these don't look too different, and therefore it's always been hard to differentiate, and therefore that's been the reason why we haven't known about this pro-reparative M2 phenotype for a very long time. And so what happens is there's this dynamic equilibrium. So initially this M1 state is required, brings about the inflammation, brings the other actors to the site of injury, and then these M2 macrophages are brought about because the equilibrium shift towards the M2 pro-reparative phase. So if for whatever reason, the macrophages remain in their M1 state, they release these pro-inflammatory cytokines, TNF-alpha and IL-1 beta, that bring about bone resorption. Now, when they shuttle to this M2 pro-reparative state, they release IL-10 and TGF-beta, which inhibit bone resorption. So this is the macrophage and bone resorption dynamics. So what about bone formation? Now here, the M1 macrophages, like I said, bring the other actors to the site of injuries, like the MSCs. And the MSCs release anti-inflammatory compounds and cause this shuttling from this M1 to M2 state. And so this M2 macrophages work in tandem with your osteoblasts and help with the maturation of osteoblasts to lay down bone. So what happens is, for some reason, if the biomaterial or the insult or injury or any sort of cues persist such that the M1 state remains, this causes an inhibition of the MSCs to differentiate into osteoblasts, and thus impedes the whole bone formation process. So this is the very, intrinsic relationship between the immune system and the bone response in biology and to a material. And so what happens is, we looked at understanding this through an experimental paradigm. We looked at the osteogenic SAOS2 cells, and we also looked at the macrophages. And I kind of quickly touch upon some of the results that we found from our macrophage study.
As seen here, rat monocytes are introduced to conventional hydrophobic PEEK on the left and negatively charged hydrophilic Z-Fuse composite on the right. The monocytes initially signal the M1 phenotype commensurate with normal wound healing and the routine initial inflammatory response to wound or injury. The immune system recognizes hydrophobic materials as foreign contamination since native surfaces within the body are hydrophilic. At day 4, a sudden and drastic shift to an M2 phenotype was observed on the Z-fused surface. The M2 phenotype signals for anti-inflammatory and cellular repair cytokines, such as interleukin 10 and interleukin 12. IL-10 and IL-12 contribute to the maintenance of bone mass through the inhibition of osteoclastic bone resorption and upregulation of osteoblastic bone formation. Contrarily, the inert hydrophobic surface of PEEK demonstrated a prolonged M1 inflammatory response, along with protracted increases in IL-1 beta and IL-6 beta cytokines, which are known to contribute to vascularized fibrosis or walling off of a foreign body or implant, deemed to be not self or dangerous to the immune system because of its hydrophobic nature.
So that's pretty much what happened on Z-Fuse versus PEEK, and we find that from the immunolabeling studies, we saw the same thing. We saw that there was M2 expression, which is the pro-reparative phenotype of the macrophage on Z-Fuse versus, say, PEEK, and we looked at the Fizz, which is a marker for M2 macrophages, and we found on Z-Fuse there was an upregulation of this M2 marker on Z-Fuse relative to titanium and PEEK, and this trend stayed pretty consistent. Also, what was very interesting was when we gave it a pro-inflammatory push, we took these materials, brought about a pro-inflammatory insult, and what happened was, very interestingly, Z-Fuse, even after a pro-inflammatory insult, took to the shuttling of this pro-inflammatory state into an M2 pro-reparative state. And so that was very impressive on Z-Fuse versus, say, PEEK, which resulted in this M1 pro-inflammatory state actually staying put. Another important aspect of the protein expression was, again, we looked at BMP7 and osteocalcin, and you could see that, again, Z-Fuse outperformed titanium and PEEK on these bone response characteristics and parameters. So in conclusion, what we were able to study through the immunolabeling and PCR analysis was there was increased M2 expression on Z-Fuse relative to PEEK or titanium, decreased IL-1 beta, TNF alpha, which are pro-inflammatory cytokines. And also we found that even with a pro-inflammatory challenge, Z-Fuse drives these M1 to M2 polarization. Interestingly, increased osteogenic protein expression was seen on Z-Fuse compared to titanium and Z-Fuse. And we believe that the intrinsic material chemistry, that is the hydrophilicity and surface charge, drives this M2 polarization. So quickly then we went on to elucidating this mechanism out in a sheep study. And we basically took sheep. We had two groups, one with PEEK. We had two levels, C2, C3, and C4, C5. So each level had a PEEK and Z-Fuse implanted in it. And we looked at 12 and 26 weeks. And so interestingly, we went about a very aggressive ovine model, which brought about in the cervical space of basically because of the range of motion being a lot more exaggerated in the cervical spine of the sheep as against the lumbar spine, whereas in humans it's just the opposite. The lumbar spine, lumbar segments have a lot more range of motion, and therefore the cervical spine was a better model and representative of what happens in a human lumbar-like situation. And also importantly, we didn't plate it. And as a result of not plating it, this would allow for exaggerated range of motion. And also this would really push the device in terms of, exceeding almost what would be seen in a clinical like scenario. So what we did find very interestingly at both 12 and 26 weeks was we found that the Z-Fuse, as seen in the in vitro studies, really reduced the interleukin 1, which is a major pro-inflammatory cytokine, which results in this fibrous encapsulation as seen with PEEK. That wasn't the case with Z-Fuse, as was seen with PEEK. And this was very important. We could see all the pro-inflammatory cytokines, IL-1, IL-6, and TNF alpha, all three were reduced on Z-Fuse relative to PEEK, and was statistically significant on interleukin 1. And also that resulted in very interesting bone response. You can see that there was osteophytic immature bone formation in the case of PEEK at 12 weeks, down there, you can see that new bone formation beautifully into the teeth of the implant was seen. Healthy looking cancellous bone everywhere on Z-Fuse at the bottom at 12 weeks. At 20 weeks, 26 weeks, look at that at the bottom. Just a beautiful fusion that's seen in the coronal plane as well as sagittal and the axial plane, all three planes. Whereas you can see that there was a fibrous defect in the case of peak and kind of fibrous encapsulation and lucency around the implant was seen. And so what we also saw was bone ingrowth that was exaggerated in the case of Z-Fuse relative to peak 26 weeks at both sites. And this basically drives home the correlation between osteoimmunology and bone response, that initially from an M0 restive macrophage phenotype, an M1 phenotype is necessary because it brings the other actors to the site of injury. But then quickly it needs to shuttle into an M2 pro-reparative state. So we need to develop immuno-informed biomaterials that help, induce this M2 polarization of macrophages, since it has a regenerative function. And Z-Fuse, to our knowledge, is the first synthetic immunomodulatory biomaterial that does this. And we believe that there's a huge correlation between surface wettability or hydrophilicity in M2 phenotype. And we also believe that implant wear debris, especially from titanium, has resulted in M1-like phenotypes, which have been documented in various scientific papers out there. So understanding this osteoimmunology and actually keeping that as an important factor in tailoring the next generation orthopedic biomaterial is going to be a very key factor in terms of predicting downstream clinical response. So in closing,
the extremely hydrophilic surface of Z-Fuse alters the surface charge on the implant, resulting in a negative ionic charge. This creates an ideal environment upon implantation to immediately attract and adhere the required precursor proteins for bone growth to the implant surface. The rapid shift at day 4 to M2 macrophage phenotypes signals the start of the healing process via early osteoblast proliferation and increased adhesion, accelerating osteogenesis and ultimately supporting long-term osteointegration.
So we've initially targeted the spine market. Like we said, Z-Fuse is approved since 2019 for lumbar applications. We're looking at other adjacencies in the total joint trauma, as well as the dental and craniomaxillofacial market very soon. From a material standpoint, with respect to titanium and PEEK, you can see that Z-Fuse offers better biological fixation, is actually osteoconductive as a result of harnessing the osteoimmunology. And it has these perfect load-bearing and biocompatibility characteristics that are needed to perform as a long-term biomaterial. So again, this is the 2018 Orthopedics This Week award that we were given in 2018. With that, I'd like to thank Matt and the OMTech group for the opportunity. Thank you.
I'd like to thank the speakers for their preparation and presentations to the audience. At this point, we'll move into a brief question and answer period. Richard, from your presentation, there appears to be many benefits of using citrate in orthopedic biomaterials. What would you say is the single most important benefit?
Thanks for the question, Matt. If I had to boil it down to one, I think the most important part would be to maintain that bulk chemistry. The citrate molecule is multifunctional and allows for that pendant chemistry to be built into the bulk of the material and not just a surface modification. So whatever modifications and whatever other molecules we're attaching to this citrate material, it's going to be throughout the whole life cycle and not just a surface phenomenon.
Thank you. Shiram, you mentioned developing a Z-Fuse master file for the FDA. How do you think that will help in future submissions for a medical device manufacturer?
Thanks, Matt, for that question. I think, especially when we're developing a platform biomaterial or a biomaterial with manifold applications, I think what's very important to consider is a master file. Generally, master files are developed by biomaterials manufacturers, and the end medical device manufacturer has an access letter through which the FDA reviews relevant sections of the master file with respect to the material. I think when oftentimes with young companies, which like us, who have a leg in both boats, both as a biomaterials manufacturer as well as an end device manufacturer. I think what has really helped is the development of this master file and this outlook from the get-go, because FDA has had a specific documentation at their end through the master file, wherein they can review a whole lot of trade secret or confidential type of information. And as well as we've managed to use that existing master file and call upon it through our different 510K submissions that are even currently ongoing. So the existence of a current up-to-date repository in the form of a master file has really benefited, because the reviewers also can quickly access different relevant sections of the master file when called upon in a 510K application or a PMA application for that matter, and will evaluate those respective sections and get back to you. And I think it has helped with making the 510K application less cumbersome for the lead reviewer. And I think that's really helped, and they've appreciated it. I think it's such an important aspect to consider, especially when you're thinking of developing a platform biomaterial with different applications that you foresee different submissions going in on the basis of that particular material.
Thank you. Mary Beth, very interesting presentation. Many people, many companies have tried to address cartilage repair. We've seen many things come and go. Why do you believe that Hyalex will be the one to succeed? And if so, what are the applications that you're pursuing?
Great question. So, you're right. There have been a lot of companies who have been active in the cartilage repair space, and a wide variety of approaches have been taken, including a lot of attempts at biologic repair, which is not right for all patients. Not all patients respond in the same way. And we find that, there are just some patients, because of their age, because of their weight, because of their activity levels or the things that they want to be doing, the biologic repairs are not right for them. And so there are other companies as well that have tried synthetic approaches too, but they've used materials that have been the traditional orthopedic materials. And what's different about Hyalex cartilage is the fact that we are building a material to act like cartilage and match those cartilage properties. And we think that's the key to being successful in the cartilage repair market. And then to the second part of your question about applications, we don't have those kinds of limitations that some of the current surgical approaches have, where there's a limit to the amount of area that you can repair. We can create our implants in a variety of sizes and shape and be able to address all kinds of different implants or rather lesions for patients. So that kind of flexibility, I think, again, will allow us to treat more patients that might not have been eligible for other types of repairs.
Thank you. Certainly in the development of this variety of materials, we've all faced challenges in bringing the product through to commercialization, dealing with the regulatory agencies. Acuitive certainly had some challenges there. There seems to be an evolving landscape of requirements and testing methodologies, and it seems like the target's always moving. I'm wondering, Shiram, if you can share any experiences you had in dealing with FDA bringing your product through to clearance.
Okay, so one thing we've seen in the case of diffusion was we began with one set of one lead reviewer, and through the course of our submission, the lead reviewers had changed about 4 times. And what had happened in that process was, like I mentioned about in my talk, we had this novel ovine cervical model, which was without plating it and had this exaggerated range of motion to kind of mimic a worst case scenario as against the standard model of taking an implant and plating it in the OMA and lumbar spine, which doesn't have that same range of motion and almost like results in a hemicorpectomy with the end plate removal, which we didn't do in our case. So what happened in our model was it was a lot more worst case. We had brought about a justification of why we were going with that model, because our zeolite is nothing but an aluminosilicate, like I mentioned, and aluminum is a potential neurotoxin. And so to look at what would be the potential adverse effects if any sort of dislodgement or movement or micro debris could come about, we had actually come up with this very innovative model that the lead reviewer at that point in time was very excited about and was totally on board with it. What had happened was subsequently with the change in lead reviewers, by the time we reached our fourth lead reviewer, this person was of the opinion that this model was not, and especially after we had seen in the case of the control peak, there was a lot of dislodgement, and therefore we had to lose certain samples as a result of them not being able to be evaluated effectively. And so what had happened was the power of the study had to drop a bit. And so she was more concerned about the statistical portion of it rather than not be too concerned about the model itself per se. And she was of the opinion that we would have to repeat the study. And having spent the kind of time and effort and money on this, it was a nightmare for us to deal with it. Luckily, what we had done, through all our meetings with the FDA, we had very detailed notes, as well as we had insisted on having the division chief on every meeting from the get-go, and having him as the continuing link through this whole process and this ever-changing process and each lead reviewer's perspective, I think really helped, because having these detailed meeting minutes, as well as having the division chief who was on board and bought into the whole process from the get-go, was critical, because the outcome could have been very different for us if that hadn't been the case right from the start. So I think hopefully that'll give you a bit of idea and a few insights as to what needs to be done from the get-go of really involving not just the lead reviewer, which is very important to establish that working relationship through the course of a submission, but also sometimes the branch chief or the division chief, who can be crucial in such cases. Thanks.
Certainly, we must all learn to persevere, and obviously we all have, as we've all lived through some successes. So with that, I'd like to thank all the speakers and thank Omtech for the session.
Thank you to our great panel for sharing their backgrounds on their materials, and thank you to those watching. You can learn more about our sponsor, Precision Medical Technologies, at premedtech.com, and you can access more expertise and insight on orthopedic innovations at omtechexpo.com.